This is CRISPR-Cas9, the ethical debate surrounding genome editing. So the structure of this presentation is as follows. I will introduce what CRISPR-Cas9 is. I will go over its modern and its ethical implications. I will discuss the risk and the reward, and I will inform my audience about how to take action. My name is Emily Stout. I am a biomedical engineering student at Florida State University. I have been studying CRISPR-Cas9 and its modern advancements and applications in the biomedical engineering field, and I also understand both sides of the ethical debate, and my goal today is to present my opinion about CRISPR-Cas9 and the ethics surrounding it. So to begin, what is CRISPR-Cas9? So CRISPR-Cas9 is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. And the latter end of that, Cas9, is actually the name of its associated enzyme. So in a sense, CRISPR-Cas9 is coined as the universal gene sequence. And what CRISPR-Cas9 does is it targets a DNA sequence. And using its enzyme Cas9, it unzips a mutation in the DNA. In other words, it removes this mutation, and it's actually able to code for a healthy gene sequence. So in a sense, CRISPR-Cas9 is able to fix genetic mutations, and it's able to cure genetic diseases. So there are many modern applications of CRISPR-Cas9 because in the past 2 decades, CRISPR-Cas9 has been gaining traction because it's the only tool of its sort. There's no other tool that is as effective or as efficient as CRISPR-Cas9 in genetic modification. So the modern applications of CRISPR-Cas9 are extremely interesting. And the first experiment that I want to inform you all about is the scientific experiment with hemophilia B. So scientists conducted an experiment with mice that coded for the genetic mutation of hemophilia B, which is a bleeding disorder. And what they did is they took the mice's DNA and they used CRISPR-Cas9 to target the mutation that coded for hemophilia B. CRISPR removed it and then coded for a healthy genetic sequence. So the results of this experiment found that CRISPR-Cas9 was very effective and very quick at its job. And it was successfully able to not only remove the mutation, but to code for a healthy sequence, which in turn led to the mice living a healthy and long lifespan. The next application that deals with CRISPR-Cas9 is research in sickle cell anemia. And the sickle cell anemia has it has more of a human application rather than the hemophilia example that I just used. And in sickle cell anemia, scientists are finding that CRISPR-Cas9 is actually a good form of genetic therapy in treating this disease. Now, they're still hesitant to call it a cure because the effects of CRISPR-Cas9 in humans with sickle cell anemia is more so to relieve their symptoms rather than to completely get rid of the disease. However, in the animal testings in the past 2 years, scientists and researchers have actually been finding that CRISPR-Cas9 is able to not only target sickle cell anemia, but it's able to remove it and code for a healthy sequence. Now, the last application, genetic blindness, is the most cutting-edge application so far. So, researchers, again, have been doing CRISPR-Cas9 experiments with blindness on animals, but they've been doing it on the embryos of animals. And this is really interesting because the results of embryonic modification with blindness is actually being translated into human applications, in that scientists are realizing that they can actually go into a human embryo, change its genes, and then implant it in a woman who then can give birth to this fetus that has been genetically modified. So this whole embryonic modification leads into the biggest ethical implications of this topic. So as I mentioned before, the ethical implications of CRISPR-Cas9 are rooted in the ethical debate around embryonic modification. So, reminder, many ethical beliefs stem from personal bias. And in this case, I think the biggest bias that arises is the belief that life starts at conception. So if life would start at conception, this would mean by that kind of logic that in doing genetic modification on an embryo is unethical because there's no way to get that embryo's consent since it is considered to be alive in that viewpoint. But this belief, again, is very ambiguous as there are many different interpretations of it, and there are many people who disagree with it as well. So, unfortunately, in the past decade, the ethical beliefs and the ethical debate surrounding CRISPR-Cas9 has led to many kind of scientific standstills, as it's very hard to get approval to do human clinical trials when there's so much ethical debate still kind of floating in the air of the scientific world. And in my opinion, I believe that ethical beliefs shouldn't necessarily prohibit scientific advancement. And with that being said, it's important to remember that ethics in itself is a very ambiguous topic, as everybody has their own set of values and their own set of personal beliefs. And when you get into science, it is very important that people trust facts over their own feelings. And with that, I get into the whole risk and reward of CRISPR-Cas9. And in my opinion, I don't believe that the ethical worry of embryonic enhancement is unfounded. I think there is great power behind that. However, I do think that science is all about taking risks and being rewarded. And to touch on the risk side a little bit more about this ethical debate, ethicists are worried that CRISPR-Cas9 will lead to a phenomenon called designer babies. And designer babies are practically just babies that have been genetically modified as embryos. And in a sense, with CRISPR-Cas9 technology, it is possible that a person could go in and genetically select the traits that they wish that their future baby would have, and simply select them, have it be put on their embryo, be implanted inside of them, and then give birth to a baby. So in a sense, ethicists are worried that this would strip the world of its sense of natural individuality, which this argument isn't unfounded at all. It makes sense. It would be dangerous for people just to be able to go and select their child's traits. However, this kind of thinking is what's causing this big roadblock in genetic advancement, because this fear is so implanted into so many people's minds that, there's going to be a human race created completely through genetic editing and genetic modification, and we're going to handpick our traits. This fear is so widespread that it's actually causing scientists to not be able to progress with their research on things like genetic therapy and genetic modification of diseases like hemophilia and sickle cell anemia, that there's no benefit to society at this point in time. And again, this goes back to risk and reward. I believe that taking the risk and hurting some people's feelings is going to provide more of a reward in the long term, when society is going to be rewarded and benefited by CRISPR-Cas9 technology. Our lifespan is going to increase, likely just like the mice did in the hemophilia B experiment, because now CRISPR-Cas9 is going to be able to not only cure genetic diseases, but it's going to be able to prevent them at the embryonic stage. So now I want to talk about just taking action. So as a biomedical engineering student, I encourage other biomedical engineering students to go into their career with a mindset that facts are more important than feeling in science. And of course, ethics is always going to be a big conversation because there's always going to be personal feelings involved. However, if we want to progress as a society, we need to make sure that we are prioritizing scientific advancement in the face of human health rather than personal ambiguous belief. And to relate this back to CRISPR-Cas9, of course, people are always going to be warranted to feel the way they do, but these feelings should never, as they are now, prohibit the advancement of CRISPR-Cas9. And if they continue to do so and they continue to prohibit the advancement, our society will be at a scientific standstill, and CRISPR-Cas9 will just be a long-lost idea that was never able to live up to its full potential. Thank you so much for listening.